Thank you very much for the kind introduction. At the outset, I would like to thank organizing committee, particularly Dr. C.P. Chaturvedi, for inviting me to this gathering to deliver this lecture. Today, I shall be talking about mechanisms of functional inactivation of CEBP alpha leading to myeloid arrest in AML. So, before I start to my key findings, let me briefly talk about what is differentiation arrest and why it is important. As you know, during hematopoiesis from hematopoietic stem cell, common myeloid progenitor and also side by side common lymphoid progenitors are formed. But because my findings are more on myeloids, I'm talking more about the myeloid pathway. So these common myeloid progenitor pathways then give rise to the megakaryocyte erythroid progenitors and granulocyte megakaryocyte progenitors, and then they subsequently differentiate to the functional cells. I don't think it is visible there quite nicely, but I think we will have to deal with it. What happens during leukemia is that because of the leukemogenic events, such as mutations or any chromosomal translocations, those blasts keep on accumulating. Here, these blast cells keep on accumulating, and once they are accumulated, the normal, completely differentiated cells are not formed, and because of this, there is differentiation arrest. And this differentiation arrest is often can be overcome by certain mechanisms, particularly ATRA differentiation therapy is there, particularly in APL cases where ATRA is given, and recently now for treatment point of view, IDH inhibitors are also there that can induce differentiation in these blasts so that these cells can be differentiated to the normal cells. Well, the standard form of therapy is the chemotherapy, but my point of showing this slide is that the key pathway of myeloid differentiation, particularly the formation of these functional cells, is regulated by two key transcription factors, and they are CBP alpha and PU1. If there is higher amount of CBP alpha, lower amount of PU1, it drives towards the granulocytes, and if there is higher amount of PU1, lower amount of CBP alpha, it drives towards the macrophages. Now, in most of these cases, it has been shown that the function of CBP alpha is impaired. So, in all those cases where there is myeloid arrest, in majority of the cases, CBP alpha expression or function has been shown to be impaired. Now, what is CBP alpha? Well, CBP alpha is a transcription factor that contains a basic rich listen zipper, which is located in this C-terminal region, and it has two transactivation domains. So this BZ domain, it binds to the DNA, all its target genes, and through its transactivation domain, it interacts with the cofactors and co-repressors and regulates number of target genes. Well, it is very important factor because it can regulate both differentiation as well as growth arrest. Through its transactivation domain, it binds with the cell cycle regulatory proteins, causes the growth arrest, and once the cells are arrested, cells can be, can exit from the cell cycle and they can differentiate to the functional cells. And there, while differentiation is taking place, it can regulate those factors that is required for the differentiation of the granulocytes. For example, GCSF receptor and CBP epsilon, these are the two key transcription factors that are regulated by CBP alpha and are required for the differentiation of these progenitors to the granulocytes. Well, CBP alpha was initially identified as a liver protein, but later on it was discovered that it is one of the key factors that is required for the formation of the neutrophils, particularly the granulocytes. Now, this was the landmark paper that was published in 1997 in PNS by Danter and group, where they showed that in a conditional knockout mice, when they evaluated all the blood lineage cell, particularly myeloid lineages, they found that even in the heterozygous, there were both neutrophils as well as macrophages, but in the conditional knockout mice, there were only monocytes and no neutrophils. So this was the first landmark study where they showed that CBP alpha has a key role in the formation of neutrophils. And down the line, it was found that in majority of the AML cases, CBP alpha is mutated. In almost 10 to 15% of the cases, there is mutations. And in many of the other cases, it has been shown that there are certain endogonistic proteins or post-translational modifications that make it inactive. Now, these are the other mechanisms how it has been shown that CBP alpha, I mean, like I said, it is very crucial for the neutrophil differentiation, and hence it is at the center of attack in most of the AML cases. Well, people have shown that in many of the cases there is CBP alpha mutations. There are also inhibitions at the mRNA and promoter level. There are promoter hypermethylations of CBP alpha, and particularly in case of CML, if you look at the BCR-ABL positive CML cases, there is CBP alpha mRNA, but there is no CBP alpha detectable protein. And people have shown one Nature Genetics paper where they showed that in BCR-ABL positive cells, there is no CBP alpha protein because the mRNA of CBP alpha is inhibited by the RNA binding proteins. So CBP alpha mRNA inhibition has been shown. Subsequently, if the protein is formed, people have shown that the post-translational modifications of CBP alpha occur that render it inactive. For example, phosphorylation at serine 21 in FLT3 ITD positive mutations makes it transcriptionally inactive. In addition, there are many antagonistic proteins that have shown to interact with it and inhibit its transcription ability. We have recently published many papers where we have shown that there are certain antagonistic proteins that can interact with CBP alpha and inhibit its function. Recently, we published some papers where we worked particularly on the protein levels of CBP alpha, and there we tried to identify what are the E3 ligases that might be involved in regulating CBP alpha protein stability. So that's what I will talk today about. But before I go into the protein stability pathway, let me briefly talk about the ubiquitin cascade. Of course, you all are aware, but I just would like to mention that ubiquitin cascade is basically a sequential action of various enzymes, E1, E2, E3, and very importantly, de-ubiquitinases drugs. So there is interplay between E3s. These are the E3s, and then these are the dubs. So what happens when a substrate, for example, CBP alpha in this case, is polyubiquitinated by these E3 ligases, and once that substrate is polyubiquitinated, that is subjected for the degradation by the proteasome pathway. However, simultaneously, there are certain dubs that keep on removing these ubiquitins so that the homeostatic levels of the protein is maintained in the cell. So there is utmost need of dynamic equilibrium between the action of E3s and dubs. So we hypothesize that if there are certain E3s that are activated during leukemia, and if they are polyubiquitinated CBP alpha in a way that its protein levels are reduced, then that could be one of the possible target through which we can reactivate or restabilize CBP alpha. So today I'm going to talk about one E3 ligase and one kinase through which we have shown that they negatively modulate CBP alpha protein levels. Now, we in the first part, we are showing that CDK2, which is very common cell cycle kinase. I mean, one paper showed that during differentiation of blood cells through ATRA, because ATRA is a clinically used differentiation therapy in AML, and in that study that they published in 2018, they showed that during differentiation there is degradation of CDK2. So degradation of CDK2 is required for the induction of differentiation is what they're finding. So based on this, we speculated that if there is CDK2, it must be inhibiting CBP alpha, or it could be inhibiting CBP alpha. To address that, we first evaluated, does inhibiting CDK2 through pharmacological inhibitors brings any changes in CBP alpha protein levels. As you can see here, when we treated with the roscovitin, which is a CDK2 inhibitor, in THP1 and U937, these are both in vitro cell lines, there was restoration of CBP alpha in the treated condition, which indicated that inhibiting CDK2 might impact on CBP alpha protein levels. We further, in U937 itself, we depleted CDK2, and we found that when we depleted CDK2, there was copious restoration of CBP alpha in this condition. So these two, kind of ensured us that inhibiting CDK2 might restore CBP alpha. To address this further, we overexpress CDK2 to see if we are getting the reverse finding. And we found that when we overexpress CDK2 here, CBP alpha expression, the endogenous expression reduced. Similarly, because CDK2 was downregulating CBP alpha protein, and like I said, we were interested in looking at the protein level. So we evaluated if this downregulation is via proteasome pathway or not. So here, when we treated with the increasing amount of CDK2, you can see CBP alpha was going down, But when we inhibited with the proteasome inhibitor, in this case, if you compare between this and this, there was restoration, indicating that CDK2 might be downregulating CBP alpha by impacting its protein turnover. Now, to further confirm this, we perform this cyclohexamide treatment assay, where you inhibit the neoprotein synthesis and whatever protein is available will degrade at its normal rate. So when we followed it up to 240 hours, we found that rate of degradation of CBP alpha was slower compared to when we overexpress CDK2. So the CDK2 expression exaggerated the degradation of CBP alpha, and this was further confirmed in also in the endogenous condition, that when we gave roscovitin, which is a CDK2 inhibitor, the CDK2 protein, sorry, CBP alpha protein was stabilized and its degradation was also restored. So indicating that CDK2 impacts CBP alpha protein levels. Now, like I said, that with proteasome inhibition, there was restoration of CBP alpha. So how it is doing this? Because CDK2 is a kinase, it is not an E3 ligase. But we wanted to confirm if it is really happening via ubiquitination. So we used here a mutant where all the lysines of the ubiquitins are mutated in this condition. So when we overexpress CDK2 along with CBP alpha, there was slight down regulation. But when we increase the amount of lysine mutant that was having all the lysines mutated in the ubiquitin, the CBP alpha protein rather stabilized, indicating that CBP alpha protein levels are modulated by ubiquitination. So this we further confirm that indeed in the presence of CDK2, if we compare these two lanes, in the presence of CDK2, CBP alpha was heavily polyubiquitinated, indicating that CDK2 promotes CBP alpha polyubiquitination and thereby it may impact its protein levels. Now we further confirm, because for an E3 ligase to target its separate substrate, it needs to interact with it. So we confirm that these two proteins interact with each other. So we perform this co-immunoprecipitation where we overexpress his CBP alpha, and in another condition we treated with MG132 to see if whatever CDK2 is present, if they are interacting with each other. So we found that in IgG there was no interaction, but in these two conditions there was interaction, and there was more interaction when MG132 was given. We further confirm that they also interact at endogenous levels. So CBP alpha, here we have used a cell line where we have inducibly expressed CBP alpha, and when you treat them with E2, the CBP alpha expression, the CBP alpha migrates to the nucleus. So we use that system, and here we showed that CBP alpha ER does interact with the CDK2. So far we confirm that CDK2 interacts with CBP alpha and apparently affects its protein levels through ubiquitination. Now, if CBP alpha expression is getting affected, does it also affect its function? To address that, we again went back to that in vitro system where we had used this K562 ER alpha inducible cell line. So this cell line is a cell line where we have CBP alpha tagged with the ligand binding domain of ER alpha, and this can be induced with the estradiol. So we use this cell system because what happens in this system, this CBP alpha ER is expressed but is present in the cytoplasm bound with the HSPs. The moment you give the E2 binds with the LBD region of CBP alpha ER size, and then it gets free from the HSPs and it migrates to the nucleus. And in the nucleus, it can bind to its target genes and it can induce the expression of those genes. So in this system also we confirmed that when we overexpress CDK2, this degradation of this protein was very faster. But in this condition, when we gave E2 here, these cells were differentiating. I mean, it is not clear here, but without E2, most of the cells were plastic in nature. In this, they differentiated and they took the neutrophil-like phenotype. And when we overexpress CDK2, again, most of the cells were blast-like. The image is not clear here, otherwise it's quite obvious here. So, taken together, so far our finding shows that CDK2 can inhibit CBP alpha expression, protein turnover, and thereby can impact its functions. Now we were intrigued to find out how it is doing it, how it is inhibiting CBP alpha protein level when it is not an E3 ligase. So while we were, Contemplating about it, we came across an article that was published in EMBO in 2008 where they showed that CDK2 phosphorylates and stabilizes SKP2. Well, SKP2 is an E3 ligase and it is a key E3 ligase of the cell cycle pathway. So we thought that maybe CDK2 is stabilizing CBP alpha, sorry, SKP2, and in turn SKP2 is targeting CBP alpha. Well, here I would also like to mention that in our previous study, we have shown that SKP2 is an E3 ligase for CBP alpha. So that we already knew that SKP2 does target CBP alpha. So we took that into account and then we first tested. Is it so or not? So here what we found that when we overexpressed CBP alpha alone together with SKP2, CBP alpha was very nicely downregulated. And in this condition, when we treated with the roscobitin, even in the presence of SKP2, there was no inhibition, rather it was more stabilized. We also used some other inhibitors because this PB for P38 and SB for GSK3 because these are the two other kinases that were reported to phosphorylate CBP alpha. So here we observed that when we inhibit CDK2, SKP2 was unable to downregulate CBP alpha. So it gave us a clue that CDK2 might be downregulating CBP alpha by stabilizing SKP2, which in turn is downregulating CBP alpha. To further confirm if it is that way, we also depleted first we because this was pharmacological inhibition. Here we depleted. So what we did is CBP alpha alone together with CDK2, there was downregulation of CBP alpha, and in this condition, when we depleted SKP2, CBP alpha was not downregulated, indicating that CDK2 indeed seems to be regulating CBP alpha via SKP2. Now, if it is so, we again used two different inhibitors. This time we used NU614, it is CDK2 specific, and then again roscobitin. So we used two different inhibitors to really confirm if it is happening that way. And what we found when we inhibited CDK2, SKP2 expression was downregulated. Even in this condition also, with roscobitin it was downregulated, but CBP alpha was slightly restored. We used two different inhibitors and we confirmed that SKP2 is indeed getting downregulated once we are inhibiting the CDK2. Now, to further show that CDK2 is indeed doing that way via regulating SKP2, here we overexpressed CBP alpha alone together with CDK2. And as you can see, with CDK2, there was phosphorylation of SKP2 at serine 64, which was already shown in that EMBO paper. So we just followed that paper. There was no change in overall SKP2 here. CDK2 was there because we had overexpressed, but CBP alpha was down. So it does confirm that CDK2 does regulate SKP2 and thereby CBP alpha. To further confirm if it is via the phosphorylation of SKP2, we also used two different mutants of SKP2, where we mutated all the possible phosphorylation sites that were phosphorylated by CDK2. And we found, particularly in this section, if you look, when we constitutively activated this SKP2, CBP alpha degradation was very faster. So this confirms that CDK2 stabilizes SKP2 by phosphorylating and might downregulate it. We further confirm, it is not clear here, we further confirm that CBP alpha, CDK2 and SKP2 are present in the same immune complex through co-IP. While it is not clear here, you can see the bands are there in this condition. I'm really sorry for this because it is not clear here. Then we further confirm that CDK2 mediated downregulation of CBP alpha was indeed via ubiquitination through SKP2. If you look at these two lanes, this and this, here there is very heavy ubiquitination where there is only CDK2, while in this condition the SKP2 is depleted, so you don't see any ubiquitination in this. While rest everything is present in both the conditions, which shows that if you remove SKP2 and if you give CDK2, ubiquitination observed in case of CBP alpha is completely abolished. So this further confirms that CDK2 by stabilizing SKP2 through phosphorylation at serine 64 is stabilizing it. Once that is stabilized, that is ubiquitinating CBP alpha and degrading. We further confirm that when CBP alpha is downregulated via CDK2 SKP2 axis, it is also impacting its transactivation potential through luciferases on a minimal PTK alpha loop. So you can see here with the SKP2, there was CBP alpha function was downregulated, and with CDK2, it again downregulated. In the last condition, when we gave CDK2 but depleted SKP2, the function was restored. We further confirmed through gel shift, where we showed that CBP alpha binding was there when CBP alpha alone was given, but with CDK2, because there was very less CBP alpha since it was down regulated, there was no binding in this condition. This is cold flow. And in this condition, when we depleted SKP2, binding was again restored here. So this all confirms that CDK2 stabilizes SKP2, which in turn ubiquitinates CBP alpha and thereby impacts its protein turnover, which is reflected in its inhibited functions, which includes transactivation potential as well as differentiation inducing potential. Now, if it is so important, what will happen if we target both, SKP2 and CDK2? So what we observed that when we target both alone, that differentiation induction potential of U937 was just 2.4 to 18, but it almost synergized when we inhibited both together, SKP2 and CDK2. If here, the differentiation CD11B increased up to 27%, indicating that targeting both CDK2 and SKP2 may lead to the robust expression of CBP alpha, which in turn may drive differentiation. This all findings we published in Journal of Cellular Biochemistry in 2020, and in the same year, another group published that CDK2 suppression synergizes with ATRA to overcome myeloid differentiation blockade in AML cells. In the same year, same month, they also published. So these two studies were going simultaneously. So it's like what we showed, they are corroborating those findings. So with this, I would like to stop here. Before that, I would like to thank my students Gatha Thakkar, who is here. This work was done by her, and for funding to the Lady Tata Memorial Trust and CSIR New Delhi. Thank you very much. I am open to questions.